Good morning, one and all. I, Arpan lecturer of Biotechnology and Bioinformatics at Dry University in Ahmedabad. Today, I am discussing about the topic of need for bioinformatics. So, first of all, today I am covering the definition of the bioinformatics, introductions, biological problems which was occur in the biological science, and the applications and the services of bioinformatics. Now, first of all, what is the bioinformatics? It is an interdisciplinary scientific field that develops methods and the software tools for storing, retrieving, organizing and analyzing the biological data. As an interdisciplinary field, bioinformatics combines the computer science, statistics and the engineering to study biological data and the process. That means bioinformatics itself is an interdisciplinary field, and it combined with the three branches, that is the statistics, then computational science and the engineering related to biotechnology. Now, the introduction to the bioinformatics. So, bioinformatics has become an important part of many areas of biology, areas like agriculture, drug discovery, medical analysis. I will inform you later about these all topics in the application part. Now, in experimental molecular biology, bioinformatics is the techniques such as image and signal processing allow extraction of the useful result from large amount of raw data. That means if we are a pre-existed data in our database, then we are able to extract that data. It's a very fast manner and use that data for the discovery of the new data. Now, in the field of genetics and genomics, it adds in sequencing and annotating genomes and their observed mutations. That means it is useful for the sequencing project. Also, it is the annotating the genome information, and the mutation are also search to bioinformatics. It plays a role in the text mining of biological literature and the development of biological and gene ontologies to organize and the query biological data. That means we are able to, by using bioinformatics, we are organizing data of the query and store into the database that will be retrieved anytime from the database. Now, bioinformatics tools aid in the comparison of genetics and genomic data, and more generally in the understanding of evolutionary aspect of molecular biology. That means we are able to get information about the evolutionary biological information from the bioinformatics. Now, at a more integrative level, it helps analyze and catalog the biological pathways and networks that are an important part of the system biology. That means we are used as a catalog for the all pathways for the and other systems of the biological things. Now, it aids in the simulation and the modeling of DNA, RNA and the protein structure as well as the molecular interactions. Now, what are the biological problems? If we are doing any type of work, we will face some type of problems. The similar way in biotechnology field, when the scientists were try to get some new information, try to develop some new things, they are face a lot of problems. Problems like, first of all, what is the role of a particular gene? That means if suppose I have a whole genome, then how can I find out the one particular gene is it is located and which place, and what is the role of that particular gene during the expression time? Because in whole genome there are more than 30,000 genes are present, so it is too tough to find out about the role of particular gene. The next problem is, does a particular gene help to cause a disease? that means all genes express and give a protein products. That protein products, if suppose gene will be the change, then the protein will be automatically change, and it will suppose form abnormal protein. It will might be a chance to cause some disease. So this is find out, it's very difficult. Now how does a drug affect a cell? Suppose I develop one new drug and I inject it into my body. So what will be the effect, or say the drug it's affect a cell or not? That will find out. It is also a difficult task in biological science. Now can we insert a gene into a corn to protect it against a disease or pest? It means against a disease or pest, we are able to insert it into the corn one new gene. Now the next one is can we design a drug to accomplish a particular purpose? That means if right now there are lots of drugs in the market, but most of the drugs will give some toxicity or say some side effects to us. So can we design a drug to a particular purpose which is only cause a disease, not give any side effect? It is the scientists are right now doing search on these things. Now can we build a cell that it's pollution, because it is a main factor of the biotechnology field. Now biological problems that computer can help, that means number of problem will be solved by the computer, or say was solved by the computer right now. So the problem like I clone a gene, is it a known gene? That means if I have one particular gene, I clone, that means I produce a replicate of that particular gene. And if it is a known gene, how can I find out it is already predicted by other scientists, or say it is the unknown gene? So I will compare with that particular gene to the pre-existed data which was stored in the database. So I will find out all information about that particular gene. Now does the sequence match, or say is the sequence any good? That means if that gene is the new gene, then how much similarity it has to compare to the pre-existed data. And the next one is does it look like anything else in the database? That means the sequence or say structure which will find out by me in future. So I will be able to compare with that structure or the sequence to my database. Next, how can I find more family members? That means if I find out something new, I compare with the family members. If I will be able to find out the family relation with the data, then immediately I will get more information about that particular new things. So I will save my time, cost, and other lots of efforts too. Now I have an orphan receptor. How can I find its ligand? That means if in my cell receptor are present, so if I can use some drug or chemical molecules as a ligand, so how it is interact with that particular receptor, it will find out through the computational analysis. Now the gene I am interested in, it was found in the another organism but not mine. How can I look for it? So if suppose I want insulin gene, for example, the one gene which is like in pancreas, isolate of cells, it has some genes which will produce insulin. But if that particular gene it's not present in mine, so how can I look for that particular gene in other organism? That will lead by the computer analysis. Next, I have a linkage to a specific region on a chromosome X. How do I find gene at that region? That means the location of that gene particular find out by me. It is now possible through the computational analysis. Now, why bioinformatics need right now? Because before 1960, there were scientists were working on the research. They were trying to lots of develop new things, but the ratio of the discovery or say ratio of the researchers were very, very low. Hardly 10 to 20 papers were published in a single year. But after 1965, first of all, the Margaret Dayhope scientists came, and they were with her organization, they were developed a number of software or computational analysis tools. So because of that, now we are able to publish, or say to new discover, more than 8,000 to 9,000 published 9,000 papers in a year. So that much of increase the level of research, or that much of increase the ratio of the developers through the bioinformatics. Now, rapid increase in the data due to genomics, too much data to characterize now through genes or protein individually. And the most important part is the bioinformatics is the smart use of the information. You know, before 10 years, all are said like, do a hard work, But now, everyone say do a smart work. So bioinformatics provide us to a smart work to do a better by using a low effort, or we will get a lot of benefits because of bioinformatics. Now, the ideally computational and the experimental biology are always partners. Now suppose compare, we have the sequence. What does it mean? That means if suppose I have a 50 nucleotide sequence, so I can able to work on that. But suppose I have 5,000 later, I will give a much effort, or say it will be a difficult for me. But if suppose the whole human genome I have, that means the 3.3 billion nucleotides of genome, so it will very difficult for me. It is impossible to work on that. So at the 1990, the Human Genome Project was discovered. At that time, number of organizations were communicate with each other, and they were the one whole organization are come with the Human Genome Project, and they divided human genome into the small fragments, and these fragments were sent to the all other organizations. Now these organizations work on that particular fragment, and they were result sent to the same organization. So this organization were collect all information from the small organizations, and they collect all information and make a documentary on the Human Genome Project. Because of that documentary, now we are able to know about the all sequences, all proteins, and all other information related to us. Now suppose in protein structure, the primary structure is the second straight amino acid chain, which is bound with the peptide bond. Now the secondary structure, tertiary structure, and the globular structure. Now it is very difficult to work on experimentally analysis by manually on these whole things, but because of bioinformatics, because of computational tools, now we are very easily do a work on such type of informations. Now, the most important part of the bioinformatics is the application of the bioinformatics. Because if we are working on any work, or if we are developing any new things, then our prime or our aim is that to develop it is the benefit for us, right? So similar way, bioinformatics has found its application in lots of areas, like in understanding the meaning of sequences. Two distinct analyses them are emerged. The first one is the pattern recognition techniques are used to detect similarities between sequences. That means if suppose I have one sequence, so I compare with that sequence to my pre-existed data. So if pre-existed data have all information, like suppose a sequence I have stored in my database related to a sequence, I have all type of data like its amino acid sequence, its structure, its function, all type of information I have. So if I find out a new sequence and compare with a sequence, if suppose I got more than 50% similarity to that particular sequence, then I have now similar type of sequence, similar amino acid sequence, similar protein structures, all type of information I will get. But now remaining 50% I will do a work to find out all other things. So I save my effort, cost, income, everything. Now the hence to infer related structures and the function. The second pattern is the ab initio prediction methods are used to deduce 3D structures. That means if suppose I have one sequence, I will able to direct generate a 3D structure by using that particular sequence. So the ab initio prediction method is the second type of method, and ultimately to infer function directly from the linear amino acid sequence. Now the next application is the sequence homology analysis, one of the driving forces behind the bioinformatics, because sequence homology, it means we compare one sequence to the other and find out more similarities to check or to get the more information about the new sequences. Like study could be carried out in yeast like lower organism, and then result can be applied to the homologous in the higher organisms such as human. That means if suppose I want to start my work to related to any field, I will start my work to the small things, then step by step go to the higher field. The similar way scientists were started work on the bacteria, E. coli, yeast, and then after they were got the whole genome of such type of organism, they compare that genome to the human genome. So what is the benefit for that? We will get more than 30, 40 or 50% information from such type of genome, because the genome is almost the similar in different type of organisms. So we are able to find out very rapidly, very fastly to the whole information related to humans. Now the predictive function, that means if I have something unknown things, then I will be able to find out or say predict about that gene or that sequence to the future of fat. Now next important part, it's a drug designing. That means this information's based approach significantly reduces the time and cost necessary to develop drugs with higher potency, fewer side effects, and the least toxicity than using the traditional trial and error approach. In traditional and trial error approach, scientists were working on number of chemicals attached with each other and make a new drugs. Now this drug, it's through trial and error approach, we are given to one animal to check the activity of that particular drugs. But in bioinformatics, we have a drug designing software. Through this software, we are add all information related to that particular drugs into the computer. Then we will get the result, or we will get the output related to its toxicity, its benefit, its number of advantages of the drug, or the optimization level of energy. We will get the all type of information related to drugs. So we can save our cost, time, and the fewer and the side effects, high potency, all are known because of that particular software. Now the medical areas, the identification of the genes that are expressed differently in affected cell provides a basis for explaining the cause of illness and highlights the potential drug targets. That means all genes it means suppose I have a human genome have contained more than 30,000 genes. All genes give up proteins. In central dogma system, gene is a raw material and the protein is the last product. So in this process, during that process, if suppose the gene will be changed by the mutation or say something adding nucleotides or any process or physical factors or anything, if the gene will be changed, the automatically automatically the protein synthesis will be the change. It means they have the lack of capacity to work functionally, and this lack of activity, so it will be able, maybe it will cause some type of disease. So that particular disease is find out because of in medical areas it is a very disadvantage to us. So now the bioinformatics aids are able to find out that particular lack reasons and check the drug targets, which is the best target for the drug to overcome such type of disease. Now, genomics and bioinformatics are now poised to revolutionize our healthcare system by developing a personalized and customizing medicine. It will help us to develop immunity from a specific disease and pathogens, also prescription of a unique combination of vaccines now, and also lead to guidance for nutrition intake and early detection of any illness. That means now bioinformatics working on to make a personalized medicine. It means right now we are in market, there are lots of drugs. If we are intake such type of drugs, we will get some type of side effects. But during the personalized medicine, the scientists know about the all information related to us. So they make a specific reason, a specific drugs which will be taken by us. So we will get completely cure such type of disease, and we will not get any type of side effects. Now the in human genome project will benefit in forensic science, then pharma industries, discovery of beneficial and harmful genes, contribute to a better understanding of human evolution, diagnosis of disease and disease risk. So human genome project, it's a very benefit for us. Now the forensic DNA analysis and the agriculture biotechnology, the bioinformatics, it's also benefit or say number of uses in the forensic DNA analysis and the agriculture biotechnology. Even in forensics, result from molecular phylogenetics analysis have been accepted as evidence too. Now the computational studies of the protein ligand interaction, that is the same related to drug designing softwares, and the knowledge of the three-dimensional structure of the protein, it will benefit us to related to retrieve all information about the proteins. And these are the diagram. See, suppose suppose I have one protein, this one, that is the unknown protein. I don't have any type of information related to this protein. So what can I do? First of all, I clone that protein. That means I copy of that proteins into the different type of softwares, insert that protein in that software. So I will get a number of outputs, yani different outputs. That means this protein is transferred in the one software. I will get the all sequence similarity related stored in the database. Another software give me information about the side chains. Another one give me information about the protein sequence. Another software give me about the information of the nucleotide sequence. Next one is check the super families, and the another one it's give me a backbone information. So if I will insert it into the different type of software, will get different type of information related to unknown protein. Now I compile this all information and transfer to the pharmacologist. Now this pharmacologist read or study this all information, and it will now start to make a new drug which is directly affect to this protein, which is cause something new disease. So now scientists are able to make a new drug which is directly affect of that protein. So this protein will overcome its synthesis, or maybe it is degraded in our body. So we will cure for that particular disease. So it is the personal example of personalized medicines. And now the different type of applications, first of all related to structural analysis, then sequence analysis and the functional analysis. Now in structural analysis we can nucleic acid structure prediction, we are able to protein structure prediction, protein structure classifications and also protein structure comparisons. Related to sequence analysis we are able to genome comparison, phylogenetic trees, the gene and promoter predictions, motif discovery, then the sequence database searching and the sequence alignment too. Related to functional analysis, metabolic pathway modeling, gene expression profiling, protein interaction prediction and the protein subcellular localization prediction. So that much of applications in of bioinformatics in the biotechnology field. So the number of software development, database construction and curation in the bioinformatics field, so it will very useful for us. Now the most bioinformatics services are the sequencing services, then the database and the management, after then data analysis and the data services. So right now because of that much of applications, bioinformatics, it save our time, it is lead to improve a number of discoveries, it is lead to develop it or predict new things which is still unknown. So on it is the smart use for the information technology. That's why we are right now use this type of technology, and maybe it will give a number of benefits in the future. Scientists are always doing a more and more get information from the pre-existed data. So might be we are able to make a whole cell or say make a whole tissue in the computer. So we are directly work on the computer and check the whole process which is till now running into the us. So thank you so much.